So our next speaker is Natasha. So Natasha is the head of the laboratory for virus, virid, and phytoplasma at the National Institute of Biology in Slovenia. The NIB is also a part of the consortium of the EURL, sorry, for virology, for viruses, virid, and phytoplasma. So Natasha is a member of the panel on diagnostic in virology and phytoplasmology. These people decided to call themselves in a short name the VIP panel. I think it comes from there. So Natasha is working with development of tests and methodology. So this session is about what can HTS bring to your laboratory. So Natasha, tell us.
Okay, thank you, and good morning to all of you. So National Institute of Biology is independent public research institute in Slovenia. Altogether, we have almost 200 employees, and we have different departments. I'm coming from the Department of Biotechnology and Systems Biology, and one of our main research topics are about plant pathogens. And within that, we have two laboratories. One is for laboratory for detection of bacteria, and the other is, as François already mentioned, laboratory for detection of viruses, virids, and phytoplasmas. Both are official laboratory, national reference laboratory, and both we are also partner in the URL. We have in the field of detection of plant pathogens, we have accreditation since 2012. Our scope is flexible. The methods that we have accredited currently are PCR and real-time PCR, and of course also reverse transcription real-time PCR. And currently we have the accreditation for 3 bacteria, 5 phytoplasmas, tomato brown rugosa fruit virus, and for a group of begomoviruses. Regarding high-throughput sequencing, we start to learn about this technique in 2012 when my colleague Denis went to the lab of Jan Kreutz, and soon after many research work in the field of virology use this high-throughput sequencing as the main tool for the research. Soon after, we also think that this technique is also very useful for diagnostics. So we collect the knowledge that we got from different colleagues from different laboratories in the frame of different of RISCO project, cost action DIVAS, in the frame of ValiTest, and we prepare our first SOP, standard operation procedure, which include Illumina sequencing of small RNA or ribosomal RNA depleted total RNA. For diagnostic work, we also developed user-friendly automatic pipeline in CLC genomic workbench, which help us for bioinformatic analysis. And first sample, official samples that have been analyzed by high-throughput sequencing is from 2015. At the beginning, we analyzed individual samples. Usually we choose samples that has unknown symptoms. And in most of those cases, these were plants showing really clear symptoms, and we found some virus particles with electron microscope. But with the target test that we have at that time in our lab, we were not able to identify which are those viruses. So in that cases, high-throughput sequencing helped us, and with this tool, we actually find several new viruses that have not been described before in Slovenia. But for our NPPO, the price for analysis of individual samples was really high, and that's why we start to think of using on the other way. And now, from 2017, we use high-throughput sequencing for the analysis of a bulk samples, and the aim of analyzing these bulk samples are to find which viruses, in addition to known one, are present in the crops grown in Slovenia. And maybe it's easy to explain this on one example. So actually, we test, this is an example of cucurbitacea plants which we tested with several ELISA tests for Slovenian known viruses. And in this batch of 7 samples, we found in some watermelon mosaic virus, in some zucchini yellow mosaic virus, which are really widespread in Slovenia. Then we extract the RNA from each of these samples, and we prepare a bulk sample, which we then analyze as a one sample by high-throughput sequencing. With that high-throughput sequencing, of course, we detect both viruses that we detect with ELISA, but in addition, we found also cucurbit aphid-borne yellow virus, which was not described before in Slovenia. So then we analyzed each individual sample separately by cucurbit aphid-borne yellow virus specific RT-PCR, and we confirm this virus in 4 out of 7 disc samples. So this is one of our examples. And last year, we got the request from our NPPO to extend national monitoring to several quarantine viruses for the European Union, those that are listed in Annex 2, Part A. You know that these are viruses that are not present in the European Union, or if someone finds it, this is really rare. So it's really difficult to get the isolates that can be used for validation of the method and as a positive control test. In addition, for many of those viruses, there are no international standards, no Euro methods, so neither national rules. And also in our lab, we don't have any target tests. So the only option was that we can offer high-throughput sequencing. And our NPPO agreed that we can this year, for example, analyze 7 bulk samples of tomato and 3 bulk samples of cucurbitacea with this technique for searching for those plant viruses that are on that list and we don't have target assay. But what they ask us? They ask us that the results should be available in time. So why this request come from them? This is because we used Illumina sequencer, and we don't have the machine in our lab because we are too small, we don't have enough samples that this will be feasible to buy. And this means that if we wish to have a good price for Illumina sequencing, we need to collect the samples and send the batch of samples together. And then again, you need to wait for the results, and it happens many times that when actually the results were ready for reporting to our NPPO, it was end of the season when the plants were not anymore in the field. So we say, okay, we will not buy this machine in our lab, but we have nanopore technology, this Minion in our lab already, and we have already some experiences how this nanopore technology works. And from one of those studies, when we compare different Minion sequencing protocols and different sample preparation types, we found that all those 4 combinations shown here works okay with Minion. However, we found that Minion cDNA PCR sequencing of ribosomal RNA depleted total RNA gave really comparable results to Illumina ribosomal RNA depleted total RNA sequencing. So we decided that this type of the protocol we use also for diagnostic samples. And I will not explain all steps that are required and are now in this our new SCP, but maybe it's good to mention just the controls that we are using. For example, each RNA extract from each sample we control with the real-time PCR for NID5. This is our internal positive control with which we are checking either the abstraction was done properly. Then I would say that the most important control for our high-throughput sequence protocol is alien control. In our case, this is Phaseolus vulgaris infected with Endorna virus, which is present in those samples in a low concentration. And the RNA from this sample we extract, and all further steps for this sample we do in parallel with our official samples. So this alien control has double function. It is a negative control to monitor the contamination, and at the same time it's also the positive control. In addition to this positive control, we have also two other positive controls for high-throughput sequencing protocol. One is the control that we add to every sample and also to our alien control at the step after DNA digestion. So this control is the positive control for all these steps from DNA digestion after that are done after. And then in addition, we have one additional positive control. This is the control that is recommended by Oxford Nanopore Technology. And this is the positive control for library preparation and for the sequencing, and also, of course, for bioinformatic analysis. On 1 million flow cell, we can analyze 5 bulk samples and, of course, all controls. And based on our experiences, we decided that each bulk sample will be composed of up to 6 samples. So we can have the results from 1 million for 30 individual samples. And results we can actually provide in less than 2 weeks. So wet lab part is done in 4 days. Then million sequencing is running during the weekend. And then for bioinformatic analysis, we need approximately 3 days. In the bioinformatic analysis, first what we do is that we check the controls, and if the controls are in the line as we make these thresholds, then we go to the next step. This is that we check for the presence of quarantine viruses. And then the third step is to check for the presence of other known viruses. And the last step, which is not real diagnostic, but a small research step, is for checking for the presence of unknown and known viruses. Regarding checking for the presence of quarantine viruses, we have really strict rules. This means that if we would detect at least one read of the virus that is on the quarantine list, then we will test each individual sample separately, so each individual sample in those bulks separately, either again with high-throughput sequencing, or if we will find any target test, we will test with the target test, or maybe we will also able to develop some new target tests at the time. An example to illustrate how we are working. Actually, last year we got a lot of samples in the frame of national monitoring that was asked that we need to test them for tomato brown rugosa fruit virus. And those samples that show some symptoms, we prepare in 4 tubes. So actually, for each sample, we take the same type of the material into 4 tubes. 2 were stored at -80, while from 1 we extract the RNA, and this RNA we test with real-time PCR for tomato brown rugosa fruit virus. From 1 we extract the DNA, and with this DNA we test with the PCR tests for detection of Begoma viruses that are on quarantine list according to the EPPO standard. Then, we actually check which 6 samples can go in one bulk. And for those 6 samples, we then actually took the RNA that we already extracted before and prepared the bulk RNA sample, which we analyzed with high-throughput sequencing. I should say that up to now, we didn't detect any viruses in those bulk samples from the Annex 2 Part A, so those quarantine viruses. So we're lucky that we're not so much work needed to them. But we detect many other viruses. For example, in the bulk of this 6 sample, we detected 5 different viruses. So it's quite useful. And of course, using high-throughput sequencing on quarantine pests and on official samples require that the test is validated. So what we did before we go to the validation, we actually use the NCBI viral database, reference database, and we checked either all our target quarantine viruses from the list that we don't have tests. The sequences of those are present in this reference database, and we found that among 8 selected, 1 was missing. And that sequence then we add to the viral database, and this is our now reference database. And regarding analytical specificity, we actually were able to get 4 different viruses among 8 our targeted that we can check this specificity. And regarding exclusivity, we used 10 bulk samples which were tested previously by Illumina sequencing and in which were no quantiviruses were detected. But all further validation, we decided that will be done just for detection of one of those viruses in the tomato leaves. So this analytical sensitivity dilution series were prepared just for one, and just for one was tested, and we found that if more than 1 million filtered reads are generated, we are able to detect this virus in a dilution of 100. And these are actually the number of the reads that was generated in all our cases by up to now. Anyway, we did this theoretical case, either if we will just get 100 filtered reads, what is our lowest acceptable output, and in that case, the sensitivity will be 10 times less. And these 10 times dilutions were then used also for checking the selectivity, repeatability, and reproducibility, just half a minute. And we found that actually we have 100% reproducible and repeatable test. And also in this calculation, we included the results of the controls. So this is my last slide. Our aim is to get accreditation just for this virus testing in the tomato leaf materials, and we hope to have the external audit this autumn, and you will see. Thank you.
Thank you, Natasha, for this interesting presentation on the approach followed. I'm sure. Don't disappoint me, please. I'm sure there should be questions for Natasha on her presentation. Yes, Joanna.
Thank you. So you did your reference database, or you used first and then created your own database?
Actually, we use NCBI database, but we just add the sequences that were missing in that reference database to our reference database. And of course, we actually we're checking for the updates each time if there is something new.
Stefan?
And the evaluation of the data is possible with the CLC workbench?
Sorry?
The evaluation of the data is possible with the CLC workbench program for your data in high throughput, as with the CLC workbench?
Yes.
Okay. Also here.
Yeah, CLC.
Yes, it's convenient.
Two questions maybe. First, the general one, do you also use HTS for phytoplasma detection?
In the research field, yes. Yes, I have one student, PhD student, she is working with that topic.
Very interesting.
Yes.
And the second one is, what are the requirements for the reference database for your accreditation? So I know you have not done the external audit, but I think at least the internal auditors
internally
would also have some requirements for this.
Yeah, actually this was internal audit we have this Friday. And yeah, actually we found that much needs to be done in addition, including this one. We need to find how to store the data, that actually it will be possible to go back to see all of those, and this actually produce enormous amount of the data, and we need to find a solution how we will be able to store all this data for 10 years. And reference database, this was the question. Actually, each time, actually, we will see if something is new and we will update this reference database. And each time we will also check this protocol with the controls, either everything is okay. So the protocol should always give the correct results.
So you have to validate the database?
Yes, validate the database, each time with the controls.
And this is linked to a discussion we had in the panel on diagnostic earlier this week. So how do you keep traceability of the changes in database, and how are you ensure that you have a curated database? So that's really where everybody is at the moment.
Also, during our internal audit, it was the largest part of the discussion, how we will actually be able to store this data that we can actually have this traceability.
I think that in the future, that would be really interesting that you guys have a discussion on this issue. But I leave this to.
Question on, for instance, in our country, they're using immunotherapy to vaccinate tomato plants. How do you deal in the diagnostic environment with viruses that are prevailing, that are really potentially masking others?
Actually, in our cases, we already have some samples where actually there was really a high amount of the virus particles, and we actually spiked one of those control, this tomato mosaic, in those bulk of the sample. And because of our rules, that we can, that enough if you detect one read, you need to do further step. Actually, we are able to detect. So in our case, in the real scenario, when actually have some really high concentration target in other samples, hopefully this will not mask the one that is present in the low concentration. We try to actually do this scenario during the validation step.
Okay, so you do this also for your alien control with coronavirus?
Yeah, it's
very delicate if you add them, that they also might prevail and mask the other.
Yeah, but alien control, we don't spike into the samples. This is the control that is run in the parallel. Yeah, we spike just RSCC in the second, then further steps.
Okay, thank you very much, I think, for your presentation.